Taxus, commonly known as yew, evergreen tree with massive trunk. It differs from pinus in not possessing dimorphic branches. The stem is profusely branched. All the branches of the taxus are of unlimited growth, form a very dense canopy structure. The leaves are linear, small, 2 to 3 cm long, spirally arranged. Each leaf is shortly stalked and possesses a single strong vein and recurved margins. The apex of the leaf is sharply pointed due to the accumulation of silica. The scaly leaves present on the fertile shoot are opposite and decussate in manner. The taxus possesses a long and well-developed taproot. The roots are deep feeders and highly branched. The common Indian species are Taxus baccata, Taxus wellichiana, seen in Himalayas and Khasi hills. The taxus shows the branch of male and female strobilus. It is the branch of male strobilus and it bears the seeds and mature seeds. Looking into the internal structure of the stem, outermost layer of the stem of the taxus is epidermis, followed by the parenchymatous cortex having some tannin-filled cells. It is followed by endodermis and sclerenchymatous pericycle. The young stem shows a ring of conjoined collateral, open and end-arc vascular bundles enclosing a distinct pith in the center. The protoxylem consists of spiral tracheids. Phloem contains sieve cells with sieve plates and phloem parenchyma. Companion cells are absent in the stem of the taxus. The cambium is persistent and develops a thick vascular cylinder due to the secondary growth. The cambium cuts secondary phloem towards outer side and secondary xylem towards the inner side. The secondary wood is devoid of resin canals and wood parenchyma. Its tracheids show uniserrate bordered pits only on their radial walls. The tracheids also show spiral thickenings. The medullary rays are uniserrate and homogeneous, but in Taxus baccata they are sometimes biserrate. The wood is strong and dense. Due to the presence of tertiary spirals, the wood is elastic in nature. Phyllogen may develop in the older stems, showing extra-stela secondary growth. Looking into the internal structure of the leaf of the taxus is dorsiventral. The upper and lower epidermal cells are rectangular in shape and thickly cuticularized. The cuticle is comparatively thin on the lower surface. The stomata are of sunken type and restricted only to the lower epidermis. They are haplochelic in development. The mesophyll is differentiated into palisade and spongy parenchyma. The palisade is generally two-layered. Only one vascular bundle is present in the midrib region. Enclosed by a distinct endodermal layer or bundle sheath, the collateral vascular bundle contains phloem towards the lower side and xylem towards the upper side. Transfusion tissue is present on both the sides of the vascular bundle. Resin canals are generally absent. The xerophytic characters of the leaf include the presence of the cuticle, sunken stomata, transfusion tissue, and differentiation of mesophyll into the palisade and spongy parenchyma. Next is the internal structure of the root. Outermost layer epidermis with long unicellular hairs followed by large parenchymatous cortex. The endodermis with a multi-layered pericycle also followed by the cortex. And the root of the taxus is diarch in nature. Metaxylem have uniserrate bordered pits and phloem bundles alternate with the xylem. Secondary growth present as seen in the pinus. Reproduction in a taxus is seen by the formation of male and female strobilus or male or female flowers. So first one is the male strobilus or the male flowers. The male flowers or male strobili are usually yellowish in color and develop in the axils of the foliage leaves. Each strobilus contains a number of overlapping sterile bracts. Some of the bracts towards the tip of the strobilus are replaced by the stamens or microsporangiopores. Each stamen is shortly stalked and has a peltate disc bearing 4 to 8 pendant microsporangia. The microsporangia surround the stalk completely. The axis of male strobilus contains a broad apex which is consumed in the formation of a stamen. The microsporangia in the young male strobilus are compactly arranged, but at maturity they get loosened and undergo dehiscence. The presence of peltate microsporangiopores is one of the most remarkable features of the taxus. A mature microsporangium remains surrounded by an epidermal layer followed by two wall layers and sporogenous tissues. The outermost sporogenous cells differentiated into tapetum. The sporogenous cells start to behave as microspore mother cells which undergo meiosis and form microspores or pollen grains. The microspores remain arranged isobilaterally or tetrahedrally for quite some time. The development of microsporangium is of eusporangiate type and is identical with that of the pinus. 4 to 8 archisporial cells develop hypodermally. They divide and form wall layers and sporogenous tissues. Female strobilus. The female strobili or the female flowers in Taxus are highly reduced that they hardly appear as cones or strobili. They arise in the axils of leaves early in the season and mature in the next season. Each female reproductive organ consists of a short primary axis having scaly leaves or bracts arranged in opposite and acquisite manner. A short secondary axis develops from the axils of upper three scaly leaves. This secondary axis bears a few pairs of scaly leaves and a terminal ovule. The primary axis functions only as a vegetative part or vegetative branch of limited growth bearing the reproductive secondary axis. Ovule. The ovule in the Taxus is oval in shape and it is an orthotropous ovule. A single thick integument is present. The integument is free from the nucellus right up to its base, forming a long micropyle. The integument is differentiated into outer fleshy, middle stony and inner fleshy layers. Two vascular strands enter the integument from the base of the ovule and reach up to its top. A ring-like outgrowth develops from the base of the integument, surrounds the entire ovule, is called the aryle or cupule. Aryle is green and saucer-shaped when young, but at maturity it is red and cup-shaped. The aryle also receives two vascular bundles, but they are very minute and rudimentary. Pollen chamber and nucellar brake are absent in Taxus. The apex of the female gametophyte changes into flask-shaped structure called tendpole. The tendpole disappears in the later stages. Generally, up to 10 archegonia develop in the female gametophyte, but sometimes as many as 25 archegonia may appear. In the young ovule, an archisporial initial develops hypodermally in the nucellus. It divides periclinally, forming an outer parietal cell and inner primary sporogenous cell. The later divides again to form many sporogenous cells, of which generally one or more start to behave as megaspore mother cell. Megaspore mother cell divides meiotically to form 4 megaspores which remain arranged in linear tetrad. Generally, the lowermost megaspore remain functional and the remaining 3 degenerate. The functional megaspore nucleus divides, redivides and develops ultimately into a multicellular gametophyte. Male gametophyte. A microspore develops into a male gametophyte. It is uninucleate at the time of shedding. Dispersal of the microspores takes place by the wind. A few microspores are taken up to the micropyle by the wind, where they are caught into the pollination drop. Through this drop, the microspores are taken up to the nucellus, where they germinate. At the time of germination, the microspore nucleus divides and forms a tube cell and generative cell. The exine ruptures and the indent comes out to form a pollen tube. The tube nucleus moves towards the tip of the pollen tube. The generative cell divides soon into stalk cell and a body cell. In the later stages, two unequal male gametes are formed by the division of the body cell. The pollen tube reaches up to the archegonial neck by penetrating the nucellus. The brothalial cells are absent in the taxus. Female gametophyte. The functional megaspore develops into female gametophyte. It enlarges in the size. Its nucleus divides by many free nuclear divisions to form as 256 nuclei. In Taxus baccata, 128 free nuclei are formed. A central vacuole develops and thus the free nuclei become parity in position. There is a centripetal wall formation in the pinnaceae and ultimately the wall of the tissue becomes cellular. Certain archegonial initials are differentiated towards the micropolar end of the female gametophyte. As already mentioned, as many as 25 archegonia develop in some cases. Each archegonium contains 2 to 4 neck canal cell and large vendor containing an egg nucleus. There is no vendor canal cell. The cytoplasm of the egg cell contains both small and large cytoplasmic inclusions and a zone of mitochondria and a lipid globule near the nucleus. Fertilization. The tip of the pollen tube present near the neck of the archegonium bursts and both male gametes along with stalk nucleus and the tube nucleus are liberated into the archegonial vendor. The functional male nucleus and the egg nucleus fuse and result in the formation of a zygote. The other 3 nuclei degenerate. As there are several archegonia in the female gametophyte, several eggs may be fertilized, resulting into simple polyembryony, but ultimately only a single embryo attains maturity. Embryogeny. The zygotic nucleus divides to form 16 to 32 nuclei. Only 16 free nuclei are formed. Cell formation starts and soon the complete structure becomes cellular. A few cells at the tip of the embryo develop into the embryo, while the cells above it elongate and form suspensor. The embryo gradually absorbs all the endosperm on development and the seed thus become non-endospermic. The fully mature embryo is orthotropous and dicotyledons. Only one embryo matures in one ovule. The Taxus seeds are covered by 3-layered seed coat. The outermost layer is thin, brown and detaches soon. The middle layer is hard and stony, while the innermost layer is fleshy. The mature seeds are covered by red-colored aryle. The aryle serves to attract birds and help in dissemination. The germination of seed is hypogeal. The seed germinates into a seedling possessing 2 cotyledonary leaves. The foliage leaves arise in the coming year. The cotyledonary leaves are larger than the normal foliage leaves. This is the typical representation of the life cycle of gymnosperm. So we can see the branch bearing the